Hello, welcome to my channel. Today I'm going to be going over the digestive system or the GI tract, and this will be covering another part of GCSE biology for AQA and the topic of organization, which is topic 2. So without further ado, let's get started. So something you might need to touch on is what are enzymes to be specific? Well, enzymes are actually biological catalysts. If you learned about what a catalyst is in chemistry, you know that is something that lowers the activation energy of a reaction to allow it to happen at a lower temperature, thus speeding up the reaction. So that's what an enzyme happens. In GCSE, you learn that the lock and key. So you know how the key is a perfect fit for the lock and vice versa. So then you've got this enzyme here, the most rubbish enzyme ever, and then you've got this substrate, you've got and then you've got this. So with the lock and key mechanism, the substrate is a perfect shape to the active site. This is what is called the active site. And then when the substrate goes inside here, it can be broken down. Oh god. So then this will fit inside and then it will turn into its two separate components, whatever that is. But then if you were to use this one, for example, then if you were to try to fit it inside, it would just not fit inside. And then as a result, there would be no catalyzation that takes place and no reaction would occur. So it's through the lock and key mechanism. Now, a few things that affect the rate of an enzyme activity is temperature. Now, if you increase the temperature, the the rate of reaction will increase. This is because particles have more kinetic energy, and then if you learn physics, kinetic energy results in particles moving faster and having more energy. Thus, there will be more successful collisions between the active site and the substance. But past a certain temperature, the rate of reaction drastically decreases, so around 40 to 45 degrees Celsius. This is because the proteins are then broken down in the enzyme, and this is because the high temperature causes the bonds inside it to break. You'll learn that in A level. But past a certain temperature, the enzyme begins to get denatured. The denaturing is essentially the shape is changing, and then the lock and key mechanism we have talked about. That means that if the enzyme's active site changes, then it will no longer be an exact fit to the substrate, and this means that you won't be able to whatever it's called react anything, and then everything will go to heck, as you could say. Another thing that determines it is pH. So enzymes only work optimally at a certain pH range. I'll actually draw the graph for temperature. So this is what a graph for temperature would look like. This is what a graph for pH would look like. So then it should be identical on both sides, because as you go further away from the optimal pH, the enzyme gets more denatured, and as a result, you won't be able to react as much and the same things I said. Another factor that affects it is the concentration of enzymes slash the concentration of substrate. So if there are more substrates, then there will be more collisions between the active site and the substrate. And if there's more enzymes, then more enzymes can react, etc. So that's essentially it. So the first thing I need to make you aware about is that there are two types of digestion: mechanical digestion, and then there is also chemical digestion. Mechanical is just physical means, which essentially means like chewing food, churning it, using stomach, etc. Which is just like chemical digestion is using chemicals like acid, enzymes, all of that in order to digest it. So now that we've got that out of the way, next we are going to be going on to this, which is the diagram of the digestive system. Yes, so this is a diagram of the digestive system. These salivary glands, or the mouth, the first thing you need to know is that this is where the food is taken in, as you already know. If you ever, the reason you chew food is to increase the surface area, and this makes it easier for lots of things to react. And then the second thing you'll need to, which is done using the teeth, obviously. The second thing you need to know is that the salivary glands produce amylase. Amylase is a form of carbohydrates, which essentially is like an enzyme that breaks down carbohydrates. So things like starch, for example, they'll be broken down into maltose here, and the increase in surface area allows stuff to be digested faster. That's why you might notice if you eat bread, for example, or a muffin that's not sweet. If you keep it in your mouth long enough, it starts tasting sweet, and this is because some of the stuff starts getting broken down into sugars, which are sugars, yeah. Then after this, it is swallowed through the esophagus, which is essentially like the food tube. Yeah, so you just need to know that it goes down the esophagus, and you don't need to know the motion that it goes down with. You just need to know that there's a muscular action and a gravitational action. So then food passes into the stomach. Now, the stomach has lots of hydrochloric acid in it. Hydrochloric acid is a really low pH acid and strong, and this allows the enzyme called pepsin to stay inside it. Pepsin is a enzyme that digests proteins into amino acids, which are their base components. And another thing the stomach does is also this is the mechanical digestion. The stomach also does the stomach also does mechanical digestion by churning the food round. So if this is the stomach, for example, and then you've got some food in here, the stomach will churn the walls so that the food gets moved around and then broken down. And that's basically it. The liver is where everything is metabolized. So you just need to know that things like urea, for example, urea is broken down. Urea is the product of ammonia. Ammonia, as you know, is very harmful to the body. So the liver takes the ammonia that forms from breaking down proteins, for example, and converts it into urea, which is a harmful water-soluble substance that just floats in the blood before being discharged by the, as you know. So then the gallbladder, that's just where bile is produced. Bile has a high pH, meaning that alkaline or basic. This has all kinds of things: carbohydrates, proteases, and lipases. All of these, the A's at the end just refers to what it breaks down. So carbohydrates breaks down carbohydrate into sugars. Protease breaks down protein into amino acid. Lipase breaks down lipids into fatty acids. And glycerol, since fats are made of these two components. So that's all what you need to know. In the gallbladder, this is made, and then the pancreas makes this, and then it is stored in the gallbladder. My bad. Yeah, so the pancreas makes all of these enzymes and it stores it in the gallbladder. Then you go into the small intestine. The acidic things in the stomach will be transferred into the small intestine, and then this bile is secreted around the start of the small intestine to neutralize the stomach acid in the food. And then the small intestine has lots of these microfolds, which are called villi. Now these villi have a high surface area in order to better the rate of diffusion of substances. They've got they're one cell thick, so that the diffusion pathway is less. They have a great high blood supply, so that substances so that there can be maintained a concentration gradient, or substances can continuously be moved and brought around. But the A level definition for you is maintain a concentration gradient. And then they also have lots of mitochondria. Why do they have lots of mitochondria? This is to do with active transport. Something you need to know. At the start of the small intestine, there is a higher concentration of substances in the small intestine than outside. So they will move via diffusion. But as these substances move out of the small intestine, then the concentration of it gets lower. And it comes to a point where around the middle to near the end, the concentration of the minerals and etc. in the small intestine is actually lower than it is outside. So normally substances will start diffusing inside to the small intestine. But active transport goes against a concentration gradient. If you want to again see it, then I've linked a video in my description. But active transport allows the minerals to continuously be moved out of the small intestine. And active transport, that's essentially why it has lots of mitochondria. So it can produce energy for active transport. Then after all of this, the waste material like fiber, etc., is put in the large intestine. So this is where all of the stuff like bilirubin, cellulose, all of that, it just stores in here. This is essentially what becomes your feces, or the very interesting word poo. So then after all of this is done, it just constantly builds up towards this, and then it is stored in the rectum, which is a sphincter of a sort. And then after you get the urge to go, for example, it's just moved out of the anus, and then it just passes, you just pass stool essentially. So that's essentially it. I hope you've found the video useful, and I ask you to like, share, and subscribe so that I can grow more and help other people. Peace.